My name is Shanae Ford, and my presentation is going to be on fluorescence resonance energy transfer, or FRET. The interactions of two proteins with one another within a cell can be analyzed by a technique called fluorescence resonance energy transfer, or FRET. It is a distance-dependent physical process by which energy is transferred non-radiantly from one excited molecular fluorophore, the donor, to another fluorophore, the acceptor. A fluorophore is a fluorescent chemical compound that emits light upon excitation. It absorbs energy of a particular wavelength and emits energy at a different but specific wavelength. GFPs, green fluorescent proteins, are widely used in order to be workable GFPs for FRET to occur. The excitation of the acceptor chlorophore, the region of the molecule that absorbs light at a specific frequency, must overlap the emission of the donor fluorophore. The proteins also must have a 10 mn or less apart in order for FRET to occur. So basically, if the proteins coupled to these GFP variants interact within the cell, the fluorescent molecules will be brought closer together, and the light emitted by the first GFP resulting in emission of the light at the wavelength characteristic of the second variant of GFP. This is why distance is a critical part in the technique. The transfer of energy from the donor to the nearby acceptor transfer in a non-radiative fashion through long-range dipole-dipole interaction. Intensity-based imaging techniques applying the method of FRET microscopy are developed to facilitate the study of interactions inside intact living cells. Because images obtained by conventional fluorescent microscopy are blurred as a result of out-of-focus fluorescence, computer approaches are applied to improve the images. Through FRET usage, scientists have been able to study protein interactions, gene expression, second messenger cascades, intracellular signaling, nucleic acid studies, ligand binding receptors, and many more. Many techniques exist. Wide-field microscopy is the simplest and most used. It is used for quantitative comparisons of cellular compartments, intracellular mechanisms, and molecular movements. Confocal microscopy allows images of increased contrast and detail to be obtained by analyzing fluorescence from only a single point in the specimen. It is used to measure visoelasticity and biochemical responses of living cells and real-time monitoring of cell membranes in natural environments. With its advantages of having controllable depths of field and the ability to collect serial optical sections from thick specimens, confocal FRET generates a two-dimensional image of a plane of focus. Multiphoton excitation microscopy is an alternative to confocal microscopy that can be applied to living cells. MP uses a tunable laser allowing excitation of a wide variety of fluorophores with higher axial resolution, greater sample penetration, reduced photo bleaching of marker dyes, and increased cell viability. In MP FRET microscopy, specimens are illuminated with wavelength of light such that excitation of the fluorescent dye requires the simultaneous absorption of two or more photons. This highly localized excitation automatically provides three-dimensional images without the need for passing emitted light through a pinhole aperture like like confocal microscopy. It does all of this without damaging the specimen. Other techniques include flip, fluorescent loss and photo bleaching, FRAP, and FLIM. FRAP techniques have become important in research in genetics, cell biology, and cancer research. One specifically deals with the reorganization of membrane proteins in T cells during immune synapses formation. T cells and antigens of T cells are immune cells and antigens are foreign substances in a host. The molecular interactions underlying regulation of the immune response takes place in the gap between the cells and antigen presenting cells. These regulated interactions are called immunological synapses. These interactions are important because without the regulation of them, the body becomes susceptible to pathogens, tumors, and autoimmunity. The experiment in the research use receptors that are organized in the synapses, and they figured out how they were joined together into functional synapses.